So the first thing that people start asking whenever we present our work is, okay, but what is RTP801? Oh, which kind of protein is RTP801? So what we know is that is an encoded protein by the gene DDIT4. It was cloned in 2002 by 2 different groups. It has this kind of structures, okay, 2 alpha helices and 4 beta sheets. It was it was crystallized in 2010 by this group. And then, as you can see here, this is the prediction made in 2024 with the AlphaFold protein structure database. So pretty much the same, without this kind of a small hydrophobic part of the protein that they had to remove to get the proper crystal. Here we can see the whole structure right now. So it's expressed everywhere at low levels, okay? And the tedious part of working with RTP801 is that the half-life is very short. We are talking about a half-life of 2 to 5 minutes. So imagine the difficulty to work with this protein. So at the physiological level, the regular functions or the best known functions of RTP801 is that is a negative regulator of the mTOR pathway, and in a second term also of the AKT pathway, because you know that mTOR2 can phosphorylate AKT. So, for example, we have that RTP801 can regulate mTOR1 via TSE1, TSE2, okay? And also RTP801 can downregulate mTOR2 and affect the phosphorylation of AKT. In our hands in 2021, what we found is that RTP801 is able to modulate synaptic transmission and motor learning. And this is important because it confirms that it has a proper function regulating protein translation, synaptic plasticity, and in the end also neuronal survival, okay? So what we know so far regarding its function in a pathological context, we know that is a stress-responsive protein. For example, it can be upregulated at a transcriptional level and a protein level in a context of A-beta, mutant Huntingtin, DNA damage, and cis-hydroxydopamine. So all these situations upregulate the expression of RTP801 or the DDIT4, and then the synthesis of RTP801. Therefore, what we have is an upregulation of their levels that what they do is to inactivate the mTOR pathway and the AKT also pathway, and everything, protein translation, synaptic plasticity, and even neuronal survival are challenged. So, what we know so far about RTP801, because we have been working with RTP801 for almost 20 years. So what we know is that in human postmortem brains from patients is upregulated in the compromised structures of PD patients, HD patients, and Alzheimer's disease patients. Interestingly, and for us, it has been really convenient because its levels are upregulated in murine models that model these diseases. For example, in the MPTP mouse model of Parkinson's disease, the R61 mouse model of Huntington's disease, and these both, the 2 models of Alzheimer, the 5xFAT mouse model and the tau model, the TG45. Saying that, this very, very short introduction, the first part of the talk, we will be talking about what we know so far of RTP801 over cognition and neuroinflammation. This work was started by Leticia Perez-Cisquez, and she investigated the relationship between RTP801 in Alzheimer's, and it has been followed up by Almudena Chicote, that her paper will be sent this next year. So at the end of this year or next year, depending on, and you will see how interesting is the relationship of RTP801 in this situation. So what we saw, or what the experimental approximation that we had, is adeno-associated virus to modify the levels of RTPA01 in hippocampal neurons in the 5xFAT mouse model. So what we found, and this is published, this is Leticia Perez's case paper from 2021, is that knocking down RTPA01 hippocampal neurons prevented cognitive impairment. And as you can see here, we did a battery of behavioral tests, and you can see here that in the T-maze, we have this prevention of this cognitive impairment, and also in the Morris-Water test, also we had this prevention of these cognitive deficits. But moreover, what we saw is that knocking down RTP in neurons also had an effect in neuroinflammation. And how we check that? We check the levels of GFAP and EVA1. And for example, here, this is a very important graph. We check the levels of RTPA01 in GFAP positive cells. So remember that we were knocking down RTPA01 in neurons, but we are checking the levels of RTP in astrocytes here in this graph. And as you can see here, between the wild types, there's an increase of the levels of RTPA01 in GFAP positive cells. But whenever we knock down in neurons RTPA01, we have a prevention of this upregulation. So this is very important to understand how we follow up this line of investigation. Moreover, we could prevent also in these mice, knocking down RTP in neurons, okay, the number of astrocytes that were upregulated in the 5xFAT, knocking down, it decreased the level, the number of astrocytes per field, and also the intensity of GFAP. So this was leading to conclude that something was going on with the astrogliosis. Moreover, we checked the levels or the number of EVA positive cells that are is a microglial marker. It's not the only one, but we have several. But here I'm showing you the results with EVA1. And as you can see here, also the 5xFAT presented an increase in microglial cells per field, but silencing RTPA01 prevented this upregulation. On the other way around, also the EVA intensity also was sensitive to the levels of RTPA01 in neurons. So we are always the same. We are checking, or we are modifying the levels of RTP in neurons, but we can see effects. We can see effects in other cells like astrocytes and microglia. Moreover, apart from these effects, we needed a mechanism. So for this reason, we wanted to check the inflammasome activation. As you can see, or I don't know if you are very familiar with inflammasome activation and involvement in neuroinflammation, that are these kind of proteins that can interact with each other, and they form these active complexes that are responsible for proteolysis or to process interleukins and many inflammatory factors. So in our hands, in this experiment, what we saw, and it was really interesting, is whenever we knock down RTPA01 in neurons in the 5xFAT mouse, we had the prevention of this upregulation of, for example, NAP1 or NAP3 or the cliff form of NAP1 that also is it can be monitored, or even the levels of procaspase-1 that was also sensitive to the levels of RTPA. So something really interesting was going on. So we decided to go down this path, since that the initial graph that I was telling you, that whenever we knock down RTPA01 in astrocytes, in neurons, we were decreasing the levels of RTPA01 in astrocytes. So here what we did was the similar experimental approach in 5xFAT mouse model, but with a different kind of adeno-associated virus. We used this kind of constructs that were specific for astrocytes. So we were sure that we were knocking down RTPA01 only in astrocytes, just because we were quite interested in what was going on, or which RTP was more decisive to mediate the cognitive impairment and the neuroinflammation. And here, for our, we were very happy with the results. We also prevented cognitive impairment, as you can see here in the TMA's results, okay, whenever we knock down RTPA01 in the 5X fat mouse model. Here, if you see me, okay, it's because we had to adapt this hRNA sequence to a microRNA because we needed to put also the GFAP promoter to be sure that this small RNA was expressed only in astrocytes. So just in case to clarify. Moreover, in the Morris water test, we had very similar results in comparison to neurons, and we could prevent also this cognitive impairment, as you can see here. Then we went to check the inflammasome activation, and we had very nice results also that we could prevent the upregulation of NAP3, the NAP1, the procaspase-1. So these are similar to the neurons results, but also we could see the levels of ASC, okay, the upregulation of the levels of ASC were prevented, okay, knocking down RTPA01. This is really interesting because it confirms that the two functions of RTPA01 in vivo model. So it's the cognitive impairment and the neuroinflammation. And in the context of Alzheimer's disease, as you may know, all of you, because you are experts, these two go together, and the feedback or the feedback loop that they get into each other is really important for the progression of the disease. Okay, so, but the next question, it arises very easily. So, okay, we know the RTP mediates cognitive deficits and inflammation, but how does it do it? Okay, so we know that we have an upregulation of RTPA01 in neurons, there are some neurotrophins in the middle, cytokines, inflammation, astrogliosis, pro-inflammatory cytokines, but we would like to know how, or to understand how these three kind of cells, they talk to each other. So for the reason to try to understand this crosstalk, we went to check the extracellular vesicles. This is the second part of the talk. This part of the talk has been done by Dr. Solana Balaguer. It was her thesis, okay? It's published. The papers regarding extracellular vesicles are published. If I explain the things a little bit fast, then you can check the papers or write me an email, no problem. I will be happy to help. And what we wanted to study is this, no? So we know that extracellular vesicles are these small vesicles that they were first described in 1971, for example, in this alga, in this Ochronomas danica, okay? And what we know is that they are membrane-bound vesicles. They can contain plenty of things that I will be talking about. For example, tretaspanins, lipid traps, immunoregulatory molecules, signaling proteins, integrins, enzymes, chaperones, RNA, and even maybe DNA. So this is important because at the beginning they believed that they were waste products of the cell, so they didn't give that much importance to these extracellular vesicles, but this has been changing over the time. So, and the good thing, but the most complicated thing is that they are released by all types of cells, okay? For the reason, we have to integrate all these signals and to try to understand all this intercellular communication. No? For example, here in the cartoon, you have a neuron, you have an oligodendrocyte, you have an astrocyte, microglia. So all these kind of cells, they release extracellular vesicles, and somehow, we don't know exactly how, but they integrate the signals, and they push through the progression of the whole life of the baby. So the previous work has been extended. We characterized how to work with extracellular vesicles. I will be talking about TVs because it's shorter. So if the first question was if we can find RTPA01 in extracellular vesicles, as you may see here, after the purification from the culture media of cortical neurons, okay, rat cortical neurons, we could see a small amount of RTPA01, endogenous RTPA01, in these EVs. So these are the regular or the mandatory markers of EVs: Alex, Flotilin, TSG, 100, and even actin. Okay, so this was encouraging, and we said, okay, so let's see if we overexpress RTPR1, that we know that is toxic for neurons. Let's see if something is changing, for example, or we can find even the ectopic RTPR1 with the GFP tag, if we can find it in these extracellular vesicles. Indeed, as you can see here from the lysates, this is with the tag, the molecular weight is 25 kilodaltons more. So instead of 30, it reaches 50. Okay, and you can see that indeed RTPR1 can be found in these EVs, in these extracellular vesicles. Moreover, it seems that the endogenous is also visible. Okay, so, but what we noticed, apart from that it was, we answered one part of the question, we answered another question that it was really interesting. Is okay, we have this overexpression of RTPR1 in neurons, but this has anything to do with the ratio of the release of these extracellular vesicles. And indeed, we saw an increase of these markers of extracellular vesicles markers, TSG 101 and Flotilin. So this led us to think that maybe the overexpression of RTPR1 was pushing the release of EVs from these neurons, these kind of challenged neurons. And we confirmed this information with nano-tracking analysis. Okay, is a system that you measure the concentration of the extracellular vesicles that you have isolated. And indeed, as you can see here, you have the control vector overexpression, and this is the concentration of EVs in this situation. But whenever we overexpress RTPR1, we increase an order of magnitude of the number of EVs released globally. Okay, so this led us to think that this was really interesting, because our hypothesis was that maybe RTPR1 could spread its toxicity via extracellular vesicles. Okay, so next what we need to prove is our hypothesis. So how we did that? We cultivate primary cortical neurons. Okay. We transduce them with these conditions, okay, a scrambled shRNA with unregulated or silenced RTPR1 within a specific shRNA. We isolated the EVs after this transduction, okay, and we send them to mass spec and proteomics facility to see which kind of proteins these EVs contain in these situations of silencing RTPR1. As you can see here, we got differentially expressed proteins in these extracellular vesicles. And when we studied the nature, what we saw, and I don't know if you are seeing this also, what we saw is that whenever we silence RTPR1, the proteins that were contained in the EVs were mostly anti-apoptotic. This is really cool. So, but to make this a solid statement, we had to go to the other way around. So what we did, okay, is to overexpress RTPR1 in the same conditions. Okay, so we overexpress a control vector, overexpressing GFP, and also the ectopic form of RTPR1 with the stack of VGFP. And as you can see here, we got plenty of proteins, and whenever we studied the nature of these proteins, most of them were pro-apoptotic. So this was confirming that at least RTPR1 was able to produce or to help contribute to spreading this toxicity from one cell or one neuron to another. But again, to confirm our doubts or this kind of a statement, we wanted to do a functional study. What does it mean? So you have the EVs, and you have to treat sister neuronal cultures with these EVs, and to check what is going on in these cultures that are receiving these EVs. So for this reason, we had some neuronal cultures transduced with these situations, silencing RTPR1 or overexpressing RTPR1 with their specific controls. We isolated these EVs, so we have the code color. We have these 4 conditions of EVs, and moreover, what we did is to do dose response curve, meaning that we treated the cells with 400 EVs per cell, or 800, or even 1600. After that, after 24 hours, we fixed the cells, and by immunofluorescence, we checked MAP2, cliff caspase 3, and the state of the nuclei, the pyknotic nucleus with hosts. To quantify these parameters, we use the cell profiler tool that we train the system to detect what was viable nuclei, or what was condensed nuclei, or fragmented, or even what was positive for cliff caspase 3. So with this tool, as you can see here, at 1600 EVs per cell, we could visualize that treating the cells with the EVs produced by overexpressing RTPA01 overexpressing neurons, we could visualize that caspase 3 was more active. Okay, we got these points, and we prepared this graph, and as you can see here, and the difference was significant between the EGFP EVs or the RTPA01 EVs. So this is the recipient cells that had more neurons with more caspase 3 active, or more cliff caspase 3. We checked also, because we are very thorough on that, we also checked the state of the nuclei, if they were fragmented, pyknotic, apoptotic, all over. And you can see here that when we treated the cells with these EVs coming from neurons overexpressing RTP, we could visualize more fragmented nuclei in the neurons that received these EVs. So far, so good. We were spreading, or we were contributing to spread the toxicity of RTPA01 from one neuron to another neuron. This was really cool. Another parameter that we wanted to check is the arborization, because is another parameter to check if these EVs can be trophic or can be toxic. So what we did, it was the same situation, okay? And with MAP2 staining, we measured the arborization of these neurons, positive for MAP2. And as you can see here, we measure the branch point, the primary dendrites, the intermediate dendrites, and the endpoints, and even the total tree length, that is the length from the soma to the endpoints, okay? And as you can see here, whenever we treated the cells with EVs overexpressing EVs produced from neurons overexpressing RTPA01, we could have less primary dendrites per neuron, or even less intermediate branches, or even the endpoints also were kind of affected. So in general, we had an effect in these parameters treating with 1600 EVs per cell. The thing that was interesting here is, since we had in parallel the same conditions, but with EVs from cells where previously we silenced RTPA01 expression, when we checked these parameters, we could see that somehow we could appreciate some trophic effect. And the cells that receive this kind of EVs, as you can see here, they have more endpoints per neuron, and the total tree length also was affected. So far, we confirm that RTPA01 mediates this transneuronal toxicity via EVs. This is the second part of the talk, and now I will start the third part of the talk regarding which kind of mechanisms, novel mechanisms RTPA01 can exert to control this protein synthesis and this viability. So I'm happy to tell you that this work was accepted in the nucleic acid research in 2 weeks ago. So the paper will be in press. So I corrected the proofs just yesterday. So if there's something, or someone is more interested in what is going on with this kind of novel function, you will be seeing this in a couple of weeks, don't worry. And this part of the work has been done by Janice Campoy Campos It was his thesis, and you will see that everything is interconnected, but is quite a cool story also. So why we were interested in RTPA1 and RNA metabolism? Because we are a protein lab, so we don't know very much about RNAs. So everything started with this result here. So back in 2016, with an attempt to understand how RTPA1 was working, what we did is an IP. We monoprecipitated RTPA1 from primary cortical neurons. We put a cross-linker for sure, DSP, okay, to assure that the interactors, the weak interactions were saved. And with these interactors, we ran these gels. We cut these bands that they were all the interactors of this protein in this situation. And we went to mass spec. Okay, we send all the bands to be sequenced and to be analyzed under mass spec. It was interesting because most of the proteins that we found interacting with RTPA1 were RNA binding proteins, and this was really puzzling us. Okay, but the next result, as you will see, it was 2 of the bands that gave us 2 different proteins, DDX1 and HSPC117. As you can see here, you say, okay, but which kind of proteins are these? No, are they interrelated? So how can you connect these to RTPA? So these 2 proteins, theoretically, they interact with RTPA1 in our hands, in these primary cortical neurons, and we wanted to go further. Digging in the background of these proteins, we saw that they were forming part of this tRNA ligase complex. This tRNA ligase complex was described for the first time in a science paper by Popov et al. In 2011. And there are other interactors in this complex, like CGI1, CGI99, sorry, Ashwin, or FAM98P. So, but what's going on with this complex? What's the function of this complex, and how can we relate with RTPA1 that we have been always super focused on the effects on mTOR and AKT pathways? So how can we connect these dots? No? Okay, so what we know so far of the tRNA ligase complex is that is involved in ligating exons, specifically the ones that comes from unsplice-free tRNAs, the ones that they contain introns. They normally don't contain introns. Most of the pre-tRNAs, they don't contain this a small part here, that is the intron. But some of them, like 7% of all the whole tRNAs, they contain this intron. And to be functional, they have to be cut here by the descent complex, and the 2 halves, they have to be ligated. Is this, in this ligation, in this part of, in this sequence of the events, that the tRNA ligase complex gets into work. So to have a proper splice pre-tRNA, you have to have this complex working, okay? Then this pre-tRNA will go to be more mature with these parts, the 5' and the 3', they will be excised, and then with all the processes involving also the cytoplasm, they will be finally amino acylated and functional to start adding amino acids in the ribosomes, okay? So, but recently, apart from ligate this tRNA half, okay, these 2 exons, it has been also this tRNA complex, it has been involved in the unconventional splicing of XBP1. Some of you, if you have been working with ER stress and the plasmic reticulum stress, you know that whenever there's this kind of stress, there's a whole answer of the cell to cope with this kind of challenging situations, and mostly XBP1S is transcribed in this transcription factor, and this XBP1S is the responsible to activate the transcription of, for example, chaperones related with ER homeostasis, or for example, calirin-7, or even BDNF, just to cope with this stress. So far so good. We have the same complex one ligating exons from the unconventional splice form of XBP1, and the other one ligating the tRNA exons. So we have two different things to check as readout, okay? So first of all, what we wanted to do is to confirm that RTPA1 was interacting with this complex, because remember that the first immunoprecipitation was in rat cortical neurons. Therefore, we wanted to check if we could reproduce this interaction in human cells, and for this reason, we use HEC293 cells. Also, we treated them with ESP, the cross-linker, and as you can see here, we could immunoprecipitate correctly RTPA1, okay? And we could check that we were in co-IP also DDX1, that is really, really good. We reproduced the HSPC117 interaction too, and even we could detect the interaction with the CGI99. So it was another effector of this complex. So this was looking good. We had an interaction that is conserved between species. So this, at least to give a relevance in the disease, in Alzheimer's disease, it can be really important. So to try to understand the function of RTPA1 interacting with tRNA ligase complex, we went back to the 5xFAT mouse model. So since we could silence RTPA1 with this adeno-associated virus, what we did is we inject again 6-month-old animals with these adeno-associated particles. We know that RTPA1 was preventing or was inducing cognitive deficits and neuroinflammation, so we know the outcome of RTPA1 in this model. We know the outcome that silencing prevents both factors or both events, okay? So from here, we collect hippocampal samples for protein to do some Western blot, but also we isolate total RNA, okay? This total RNA would be helpful to check the mRNA for XBP1 and then to try to isolate the small RNAs to sequence for tRNAs, okay? So is what we did. So for the long RNA fraction, more than 200 nucleotides each, this was for qPCR, okay? The small non-coding RNAs, with the range of nucleotides, what we did is we even enrich this fraction for a smaller tRNA or the smaller RNA. So we isolated the fraction from 60 to 100 nucleotides with an acrylamide gel. And with this fraction that it's supposed to be enriched with tRNAs, okay, we send it to the CRG and we did hydro-tRNA-seq. And also we kept some to do some functionality studies. Therefore, the first part of the talk, or the first part of the results, will be regarding XBP1. So what we are interested is, okay, let's see if RTP1 is messing up with this part of the signaling. And for sure, by qPCR, what we observe is that we measure the transcripts of XBP1S, the splice form, and also the transcripts of the unspliced form. So to see the efficiency of the splicing, people express the results by a ratio, the splice versus the unspliced form. And as you can see here, the unspliced form was sensitive to the levels of RTPA1, and whenever we did the ratio, also the ratio was sensitive to the presence of RTPA1. So we could prevent these results by silencing RTPA1. We are always with the same experimental groups, the 4 experimental groups. Wild type, SH control, wild type, SH RTPA1, 5xFAT, and 5xFAT, SH RTPA1 in a code color manner. So, okay, this was good, but it seems that RTPA1 was affecting the activity of this complex, but we weren't so sure and say, okay, let's go to check the levels of some targets of XBP1S that we know that we have BDNF, SEC24, Calidin-7, and we see. Let's see if we are doing any effect by silencing RTP-A1. And indeed, what we saw that in these samples from the hippocampal region, whenever we silence RTP-A1, what we saw is that we had an elevation, okay, of the expression or the transcripts of BDNF. And BDNF, we know that is one of the main targets of XBP1S. So it seems that it was related. And what is more important or more interesting here is that it seems that the effect of RTP-A1 over XBP1S and BDNF is pretty specific because we check SEC24D and Calidin-7, and as you can see, neither of them was sensitive to modifying the levels of RTP-A1. So we were pretty happy because, you know, BDNF can explain plenty of things at the synaptic level, neuronal survival, and all these processes that are important in a disease context like Alzheimer's disease. So the next part was the tedious part because our rationale here was, okay, if RTP-A1 is messing this complex, how can we check the effect over the pool of tRNAs? No, so for this reason, we needed to do this kind of approach, this tedious approach, that is the hydro-tRNA-seq, and from the long RNA that you can see here, that is the most of the RNA that we extracted from the hippocampi. Okay, the small non-coding RNA represented like 13% of the proportion, and in this proportion, what we have is like 38% of this small proportion is the one that is enriched or is the tRNA contribution to the small non-coding RNA. So we are dealing here with not very favorable numbers. That is what it is, and people work with that and say, okay, let's push forward the project and cross our fingers. Okay, so the first thing that we did is to measure if between wild type and 5xFAT mice we could observe any mature tRNA differences. No, all the pool you know that we have plenty of tRNAs. For different amino acids, okay, and what we saw, as you can see here, with the amino acid and the anticodon of these tRNAs, that most of them did not present any differences between wild type and 5xFAT mouse in a control situation. Okay, and say, okay, maybe it's something that. Checking the mature tRNAs, we don't see anything. So, but you know what? Since it's the ligation process, let's check the previous step. Let's check the pre-tRNA step. Okay, so we measured and we compared for each pre-tRNAs if we had any differences between wild types and between 5xFAT. And as you can see here, we had some significance and some accumulation of pre-tRNAs, mostly all in the 5xFAT mouse. So this was really encouraging. I say, okay, let's dig deeper on that and let's see if these pre-tRNAs comes from these pre-tRNAs that contain introns, and as you can see here in these red arrows, most of the ones that they got accumulated is the ones that they contain this intron that has to be excised, and then the half they have to be ligated. So we know that RTP-A1 is upregulated in these mice, and we are checking that something is going on because these pre-tRNAs are accumulated. Okay, so, but to confirm our suspicion, we had to deal with silencing RTP-A1. For the reason, what we did is to compare the pre-tRNAs that were significantly upregulated in a situation where we knock down or we silence RTP-A1 specifically. And as you can see here, for example, we have these three pre-tRNAs that were quantified. Okay, they have the anticodon, the TCT, all of them. Okay, and they will be amino acylated with an arginine. And as you can see here, they get accumulated, but whenever we silence RTP81, the tendency and the significance is to prevent this accumulation. So this situation was really clear with arginine, but also was clear with isoleucine and leucine and the tyrosine, okay? And in red, we add some pre-tRNAs with no intron as an internal control to see that the tendencies of the accumulation or the prevention of the accumulation was also fine, okay? So, okay, our next question was, okay, we know that there's something going on with this accumulation of a specific pre-tRNAs, but this can be translated to the accumulation of mature tRNAs. And as you can see here, no. We checked the mature forms of the same tRNAs, the pre-tRNAs that you saw before. Here are the mature form, and we couldn't visualize these differences that this is because it's the one with it was significant in the first previous slide, and no affectation of the mature forms. This could be a little bit of I don't know, something that it was quite weird to try to explain the functions, the novel functions, but we checked the bibliography and plenty of people, plenty of authors that were visualizing problems in the accumulation of the pre-tRNAs also saw that the mature forms of these tRNAs were not affected. And this is the system that has the cells, the super fine-tuned regulation of the cells to regulate all the translation machinery. So, importantly, okay, we functionally we needed to characterize the effect of these pre-tRNAs or this fraction of small tRNAs. Okay, these the ones that we have been working with, and we decided, okay, let's do a functional study on that. So let's get these tRNAs that contain the pre-tRNAs that got accumulated from the 4 different experimental groups, okay, the wild types and the 5X fats, with or without DSH RTP01, and let's transfect neurons and let's see if these fractions are toxic or what or any other function that they can have. And as you can see here, we also use the cell profiler to quantify all these things. And first of all, what we saw is that the transfecting cortical neurons with these fractions of tRNAs were not toxic at all. We didn't see an increase in the condensed or fragmented nuclei of the recipient cells, okay, of the transfected cells. Or even the caspase-3, the cliff caspase-3 levels were not affected also by these different tRNAs, okay? And here is the cliff, the caspase-3 intensity was not affected, significantly affected, okay? But as we did with extracellular vesicles, we checked the arborization of these neurons, no? Because we had the staining with MAP2 and we could do the skeleton of these neurons and measure, as always, the branch points, primary, intermediate, and the endpoints, and the total tree length. And here is where we had some differences. As you can see here, whenever we transfect neurons with the tRNAs from wild type shRNA, 5X fat or 5X fat with RTP01 silencing, we could see, as here, that we had differences, an increase in the intermediate branches, that it was prevented whenever these tRNAs came from the silencing situation, or even in the endpoints. And this is really interesting in the total tree length, that it will be like this, okay, that we had this difference. So it was an aberrant arborization of these MAP2 positive cells that could be prevented whenever the tRNAs came from brains where we previously silenced RTP. So far we proposed this model and the reviewers liked it, okay, and in physiological conditions, we have the active tRNA ligase complex, we have low levels of RTP, and there's a regulatory function for the proper ligation of the pre-tRNAs and the XBP1Fs, okay? But in a pathological condition, like can be in this this 5X fat mouse model of Alzheimer's disease. High levels of RTPO1 can mess with the ligase complex, okay, the ligase function, and we can see reduced ligation of these pretty RNAs or even the reduced ligation of these XBP1s. This is really important because whenever we design any molecule to inactivate a RTPO1 detrimental functions, we have to keep in mind that apart from inhibiting mTOR and AKT or inactivating mTOR and AKT, we have to keep in mind that RTPO1 can also have this supernovel function that I have been explaining. So far so good. Here is to thank all my collaborators, the usual suspects here in the University of Barcelona, Albert Giral, Esther Pérez Navarro, Eulalia Martí, the people from MASPEC in Navarra Biomed, Kike and Joaquin, the tissue bank that is the Dr. Molina, Ana Guisado, also Adrian and Luis, the RNA guys, okay. Former students in my laboratory, Dr. Nuria Martin Flores and Dr. Marcel Canal. And here is my outstanding and marvelous team that I only have nice words to describe them because they are talented, outstanding, super hard workers that are Letty, that she is a postdoc right now in UK. Janice, that defended the thesis in last June. Almudena, that she will be defending at the end of this year. Julia, that defended last October. Paul, that he's doing really cool work in following up these projects in the context of neurogenesis, and the super collaborator and super good advisor that is Dr. Albert, that we are in his cybernet group. So now, if you like, you can ask me the questions that you think that can be interesting. Thank you very much for your attention.
All right, thank you very much, Cristina, for a very interesting presentation. So the paper is now open for discussion. If anyone is interested in posting a question to Cristina, please do so by using the raise hand option in your screen, or just by sending a message through the chat stating that you're interested in making the question. I will give you the floor to do that. So while we wait for the first question, let me remind everyone that CyberNet webinar series are held every other week on Thursdays at 3:30 p.m. And the full schedule from now until the end of the year is already available at the CyberNet webpage. So if you're interested in looking at the schedule, please go to the website. Also, if anyone is interested in participating as a speaker in this webinar series, please send an email message to either Teresa Iglesias, who is the coordinator for training activities, or to myself, and I will do our best to accommodate a proper slot for you. Okay, so any questions?
Too much information.
Yeah, I don't see any raised hand. Let me check the chat just in case. No messages coming through the chat as well. Okay, so please don't be shy.
I convinced them that RTPO1 is really cool.
Exactly. Yeah. All right. So as a Cristina, yeah, I think we have one raised hand here from Amaya Hernandez. Please, Amaya, go ahead. Make sure you have your microphone on, please. Okay, go ahead.
Hi, can you hear me?
Yes.
Yes, I have one question. Do you think that both functions are somehow regulated like the new function you found compared to that of mTOR?
That is a very good question. I don't know the which contribution is worse in a context of Alzheimer's, but it can be really good to study that. I don't know how we can study that. No, I think the most important thing is to do some RTPA1 mutants and the ones that interact or mutating the regions that are responsible for the AKT-mTOR inactivation, and the other ones with the interacting with the complex. So this could be a way to go, but it sounds really complicated.
Yes, no, I was just wondering because it's really different things. So I was wondering if both take place in Alzheimer and which one is more important.
Yes, we will be studying plenty of stuff, but this is something, since we are now designing some inhibitors for RTPA1, that Paul is doing that. That is something that we have to keep in mind, because depending on the molecule where it is designed to interact to the protein, you will be inhibiting some functions and not others. So this is really an excellent question. So hopefully in a few years we will keep you posted.
Great, thank you.
Thank you.
All right, thank you both. Let me see, I think we have a question through the chat. Ready for you, Cristina.
Okay.
So it comes from I don't know where it comes from. I'm sorry about it. Cristina Ortega says, thank you for your talk, really nice. My question is about the EVs. Did you check the role of EVs from RTPO1 positive astrocytes?
The thing is, so far our cultures were primarily neurons. However, now we have implemented a culture that contains neurons, astrocytes, and microglia. So what we wanted to do right now is to see if you modify the levels of RTP in a neuron or in an astrocyte, how the content of the proteins in the EVs can change, and if the toxicity can change. So that is a really good question. So we will see. We will see. I have no idea. There are some work with if I don't know. I have no idea. So far, whenever you overexpress RTPA1 in astrocytes, it seems that it's not killing at all the astrocyte. The neuron is more sensitive. So maybe these EVs maybe are not so toxic, but maybe are not so trophic. So it's something that it can be really interesting to investigate. So thank you for the question, because it's really cool. So.
All right, thank you both. Any other question? So let's see now that the ice has been broken.
Yeah,
if there's anyone. I don't see any raised hand. No, not yet.
Not yet.
Nothing else coming from through the chat. Okay. So so let me give you the last chance to make your questions to Cristina.
I don't bite.
Yeah, I'm sure. So yeah, don't be shy. If you don't want to make the question yourself, just write them down through the chat, as Cristina just said. Cristina Ortega, I mentioned, I said. But nothing is coming. All right. So if there are no more questions, then I guess we have to finish the webinar here. But not before thanking once again Cristina for a wonderful presentation, for her willingness to share her work and her team's work with us. Thank you everyone for attending, and I hope to see you again in the coming weeks in another webinars. All right. So have a nice afternoon, everyone. See you around.
Okay. Thank you for
Bye.
having me. Thank you very much.
Sure. Thank you.